Hi micro folks, this is Professor Karen Carberry-Go. I'll be your Biology 440 General Microbiology instructor. We are in this really wild time of history with this all sheltering in place because of the pandemic coronavirus. Consequently, for fall semester 2020, as you all know, we'll be doing our microbiology class, both lecture and labs, online. And doing lectures online, I'm okay with that. But it's a real challenge for us to try to replicate the hands-on experience you would get in the face-to-face microbiology lab. So I am going to be doing a little bit of experimenting this summer, doing our online labs. I'm trying to find some high-quality virtual labs for us to do. But in addition, I'm going to kind of take a different approach to how we would normally do the microbiology lab. And what I'd like to do is actually use more history, like how microbiology evolved, how some of the lab techniques evolved. And I'll be showing you, like what some people might say, home microbiology or do-it-yourself microbiology. But I have to stress, you guys, that what I'm showing you, some of the home microbiology, be really careful. I cannot advise you to do it yourself if it would involve you potentially tasting something that might have a foodborne pathogen present. We can't have you do anything at home that would create any danger for you, your family, or your pets. So most of these will be demos, and again, I'll with most of them, I'm going to say, don't test this. If you run this little, experiment at home, be very careful that you don't swallow anything, that your children don't swallow anything, that your pets don't swallow anything. And all of the experiments, if it involves food, we need to make sure that we double bag it and we dispose of it in the garbage can. So I just want to state that, you guys. For a lot of the online microbiology classes, they're selling kits that actually have microbes that you'll be growing at home. And one of the big problems with that is that when we're growing our microbes, we call it amplification. We're getting them to increase in number. Even though in most of those kits, the microbes are what we call low virulence or opportunistic pathogens, meaning if you have a good, healthy, robust immune system, in low numbers they shouldn't cause any harm. But we call them opportunistic pathogens in that they will take the opportunity to cause harm if your defenses are lowered, or potentially if you're exposed to a really high number of them. Your normal defenses can't take care of them anymore. And that's why I'm really reluctant to have people buy these microbiology kits and be growing microbes at home. I really think for your first course in microbiology, if you're doing any of this amplification, it should be under the direction of an experienced microbiologist. So again, a lot of this will be demo, but hopefully we can relate a lot of the demos to what's going on in our world around us, what we see in nature, some of the foods we eat, some of the traditional methods we prepared foods. But I just need for you to be really careful. Don't eat any of this stuff. Make sure your kids, pets, unknowing family members or friends don't have access to it. Okay. So folks, your online OpenStax microbiology book, the first unit starts out with a section on what our ancestors knew, and I think this is so fascinating. Very often folk medicine is kind of belittled in Western medicine, but I think the process of how our ancestors observed and discovered the activity of microorganisms still has a lot to teach us. So what I thought would be, just to give you a real simple example of what our ancestors might have observed, how they were observing microbial phenomenon, even kind of harnessing them for their benefit without even knowing microbes existed, yeah? And so I thought, what's near and dear to my heart? Food, right? If I can find a food analogy for everything in this course, I'll be really happy. So I was going through our refrigerator, and what I pulled out this morning are some blueberries, which sadly I noticed they're getting soft and they're getting moldy. And I see these little kind of light white threads, these little filaments of fungi, molds are starting to grow on them. So I know a lot of fungi make toxins called mycotoxins, so I don't want to eat this. Normally what I would do is I'd put it in our compost pile where microbes would help recycle the nutrients, and then I would use the compost in growing other plants, right? So decomposition is a microbial process. But in the process, sometimes the microbes might produce some toxins that if we were to eat them, we would get sick, right? We know instinctively we don't want to eat spoiled food because it would make us sick. And then I also found some grapes. And these grapes, they don't look quite so bad. I mean, you know, if I was feeling brave, I might go ahead and eat them. But I thought, you guys, this is so cool because here right off the bat, we see microbial processes that our ancestors would have understood. The decomposition or spoilage of food here with these moldy blueberries. And these grapes, you know, they're still looking pretty good. So what I thought we would do is run an experiment in that not only were our ancestors aware of decomposition of food, but they were also aware that sometimes when food got old, under some conditions, it wouldn't spoil. The taste would change, the texture would change, but when they consumed that what we're going to call fermented food, they didn't get sick. And indeed, sometimes they found the new flavors really appealing, yeah? And so I thought I would use one simple example, you guys, of a possible fermentation. Let's pretend we're way back in the days before refrigeration, way back before we knew the existence of microbes. And let's say maybe during the fall, we harvested lots and lots of grapes, right? Well, in the old days, especially when there was no refrigeration, you'd have to try to store the plants that you harvested, say in summer or fall, to try to help you survive through the winter, right? So what if the grapes were stored in some container? And of course here, this wouldn't be a plastic container. It might be a jug or maybe an animal skin, right? And maybe as the winter went by, you know, the grapes, well, maybe they're starting to break down a little bit. And maybe if they're in like a big storage container, maybe the weight of the grapes is crushing some of the grapes on the bottom, right? So what I'm doing right now, you guys, I'm just crushing these grapes to release the juice, right? So what we're going to see is if we can recreate how humans discovered wine production, right? Okay, and we'll get into more of the details in a little bit here. Okay, so folks, just recognize these grapes, these blueberries, obviously they're not sterile, right? When they're growing out in nature, they're going to be coming in contact with microbes. There'll be microbes on the outside of them. And with these grapes, you guys, and I should have done this ahead of time, and I apologize. With these grapes, and I'm going to use my hands here, you guys, because again, we're pretending like we don't know about microorganisms, we don't know about sterile technique. I wish I had shown this to you earlier, guys, but if you get from the store grapes, you'll often notice there's a little kind of a cloudy coating to them, and I don't know if you can see that or not, you guys, that little cloudy coating. Well, in that little cloudy coating, there probably will be what we call wild yeasts, right? Like the yeast you use to make leavened bread, for example. So what I'm hoping is that we have some wild yeast on the outside of these grapes, and as I'm squishing them to release the juices, those yeasts are going to get mixed in with the juice. Can you see the juice there? Okay, so we're making grape juice. And what I'm hoping is those little yeasts will take the sugar from the grape juice, chock full of sugar, and through a process called alcoholic fermentation, convert the sugars in the grape juice into ethanol, a 2-carbon alcohol, and carbon dioxide, right? So if we're successful, you guys, maybe in a few weeks, we'll come back and check out our fermented grape juice, and we're gonna ask ourselves, has it spoiled, meaning spoilage microbes, consumed, kind of rotted, decomposed our grape mix here, or did the wild yeast, the Saccharomyces, did they win out? Were they able to outcompete those other microbes and use the sugar to make alcohol and carbon dioxide? And you guys, the cool thing is the alcohol, it will inhibit, it will stop the growth of those spoilage microbes. So this was a way in the old days and even today where we could preserve the nutritive value of the grapes without refrigeration by getting the yeast to make alcohol. The alcohol inhibits the spoilage microbes so the food wouldn't spoil. I'm so excited. Okay, now folks, what we're going to do, I was thinking maybe isn't the best way to do it, but we'll just throw it in there. So we're going to mix a little bit of the old times and present times because I thought we could do a little bit of an experiment with this. So what I'm going to do is try to sample the grapes, and I'll sample the blueberries, and we'll try to get some idea of which microbes are present. Now again, you guys, I got so excited. I should have done this ahead of time. I should have grabbed a grape before I crush it, but I think this will still work. All right, so what we're going to try to do is grow the microbes that were on the grapes and grow the microbes that are on the blueberries, right? And the way we're going to do that is we're going to use a solid food for our microbes to grow on. And in microbiology, solid media is usually named after the solidifying agent. And we'll learn later the most common solidifying agent in microbiology is called agar. It's an extract of algae. And agar is so cool, you guys, because most microbes can't break it down. 3 today. Agar, our movie here is stalling out. And I don't know if it's the heat or just the age of my computer, but hopefully this video will still be okay. All right, so you guys, what I'm going to do, I'm going to use 2 different types of media, right? And the first media is called tryptic soy agar, or TSA. This is considered an all-purpose media. It should grow a wide range of bacteria and fungi. And remember, yeast are fungi. And so you guys, this is going to be really typical if we were working together in the microbiology lab. What we do is our agar is after it's autoclaved and it's sterile, while it's still hot and melted, it's poured into a petri dish. And the petri dish has a lid, which is the wider diameter, and a bottom. And the bottom is where the agar is poured. And then we let the agar solidify. So it's a little bit like kind of really firm jello, almost that consistency. And what we're going to do is we're going to transfer our microbes to the surface of the agar. They have to be in contact with the agar so they can absorb the nutrients and use the nutrients to grow and divide and divide and divide and divide and divide. So in a few days, we should be able to look at the surface of the agar plate and see these little distinct dots, and those dots are called colonies. And they're basically a collection of little genetic clones of the first microbe that landed on the surface of the plate. And we're going to see different types of microbes make different types of colonies, right? Labeling, you guys, in microbiology is really important. So on this particular plate on the bottom, we always label the part of the container in which the microbes are growing. So with agar plates, we always label the bottom. I've put what my medium is, so I put TSA. I put day 1, right, because we're going to be sampling sequentially day after day, and I put the actual date, and here I put 6/30/20, and it's 6/3/20, getting a little ahead of myself here. And then I always put my name, right? And then I'm splitting the plates into 2 halves, you guys. And what I'm going to do is I'm going to put blueberry microbes on this side of the plate and grape microbes on that side of the plate so we can compare the 2. And then, folks, the other thing, because we're interested in seeing if we have any yeast present, I have a second type of medium. This is called Sabarod's dextrose medium, and it's selective for fungi. So I'm hoping if we have any yeast present, they're going to be really happy growing on this plate. And the advantage of the Sab agar is it'll stop the growth of a lot of environmental microbes. So hopefully we have any yeasties in there, they'll grow really well on the Sabarod's dextrose medium. So you guys, how are we going to do this? What I'm going to do, this is, again, you guys, we're going to pretend like it's caveman day. We didn't know about aseptic technique or any of that. So what I'm going to do is I'm going to grab a grape from my grape gamish here. I'm going to try to grab one of those that still has that kind of cloudy coating that I'm hoping is will contain some of our yeasties. And you guys, this is so low tech. I'm just going to roll the grape over the surface of the Sabarod's dextrose plate, and I'm going to discard this. I'm going to compost it in the garden, right? And then I'm going to grab another one, and again, trying to hopefully have that little musty, yeasty coating on it, and I'm going to put this one on the TSA plate. Now remember, on the TSA plate, yeasts and bacteria should grow. On the Sab plate, just the yeasts and other fungi should grow. And again, I'm going to compost this in our garden. And then, folks, I'm going to do the same thing with our blueberries. And remember, the blueberries were starting to look moldy. I was starting to see these white filaments, which suggests molds. Molds are multicellular fungi. So I'm going to do the same thing. I'm going to try, oh, here's a nice fungusy blueberry, you guys. That's a good one. And so I'm going to dot this one on the SAB plate. And again, the molds, because they too are fungi, they should grow on this plate. And I'll do the same thing on the TSA plate. And again, on the TSA, both the molds, yeasts, and environmental bacteria should grow. Okay, so you guys, in transferring the microbes, I really got into that one. we say that we've inoculated our medium, we've inoculated our agar plates, and because it is so hot here, it's like 103 today. If we were in the microbiology lab, normally it's not that hot, like 103, right? But because it is so hot here, we don't have air conditioning. I'm just going to hold these agar plates. I'm going to tape the bottoms to the lids, right, to make sure the lids don't fall off. And I'm going to invert them. I'm going to turn them upside down, and I'm going to incubate them. I've set up a little corner of our garage as our little do-it-yourself microbiology lab. So I'm just going to incubate them in our garage. And my only concern is it might get too hot for them. You know, ideally we'd be incubating at around 30 degrees Celsius, 37 degrees Celsius. So I'm just hoping it won't get too hot in our garage. I'll try to check them every day. And then, folks, I'm just to make sure I'm processing them the same way, I'm going to take our blueberries, right? And I'm just going to do the same thing, make blueberry juice out of them. Okay. Oops, are you still there? Okay. Make a little bit of blueberry juice out of them. Right? And again, just trying to recreate what might have happened in the old days when, say, the fruits were harvested in the summer or fall and kept in big containers, maybe animal skins or in maybe clay jars. And as they age, and maybe as the weight of the fruit on top might crush the ones in the bottom. So we're just kind of trying to recreate that. And then, folks, the other thing I'm going to do is I'm going to try to seal these plastic bags. And the reason is I'm going to try to get all the air out. And the reason I want to seal them is if we do get, say, an alcoholic fermentation going on, remember we said one of the products is carbon dioxide, a gas. So it would be fun to kind of do this maybe in a balloon, right? So if we get an alcoholic fermentation going on, there should be large amounts of CO2 gas that are produced. And so consequently, I would predict that my plastic bag would inflate over time, right, if CO2 is being made. So there's this one. Maybe what I'll do is wrap it with a couple rubber bands, yep. And I'm hoping they'll be airtight. And this is my grape juice, right? Possibly going to be wine. And again, folks, what we're going to see, remember how the blueberries were already moldy, they were already starting to spoil. It looked like the grapes weren't, right? It didn't look to me that they were moldy. So we're going to see if we can preserve the nutrients in the grapes by hoping those Saccharomyces carry out their alcoholic fermentation, right? Make the ethanol that will inhibit any spoilage microbes, make the CO2. And again, I'm going to try to flatten it, so we'll see if we get CO2 produced. And then in contrast, you guys, the blueberries, where the spoilage microbes are already kind of winning the day, we'll see if we can get alcoholic fermentation going on with the preservation, right, of the nutrients, or if we'll get spoilage. And probably the easiest way for us to tell if our blueberries are going to spoil is by smelling it, right? I think there's been natural selection so that we know when we smell spoiled food, we are repelled by it, right? Natural selection, it helps in our survival. Okay, folks, so I think that is it. One thing I'll do when the movie's over, because it's getting a little bit long, is I'm going to take the pH of the blueberries and the grapes using pH papers. So much fun, you guys. pH indicators are so much fun. And then I'll record those on our plates, and then we'll check them tomorrow. I'll check the plates. Probably what I'll do, you guys, is the next movie I'll do is when I actually start seeing growth on the plates, because that would suggest we're getting pretty good growth in our grapes and our blueberries. And then we will reculture, and then we'll re-pH, right, and see if we can see any changes that are going on. Okay, you guys, I hope you like this. I want to call this like backyard microbiology, right? Okay, and so we'll just be doing a number of these little films, both for lecture and for lab. Okay, you guys, take good care.